Hi everyone and welcome to the renal system part 2. In this section we'll be looking closely at renal physiology. So there are three main processes that we look at. The first is glomerular filtration, and this refers to water and most solutes passing from the plasma in the glomerular capillaries into the glomerular capsule within the nephron. The second is tubular reabsorption, and this is the process by which mostly filtered water and useful solutes are reabsorbed by the tubule cells of the nephron and returned to the blood. And the third process is tubular secretion, and this is the process by which waste substances are removed from the blood and are secreted into the tubule fluid. So firstly we'll look at glomerular filtration. Approximately 180 litres of fluid enters the capsular spaces each day. This is about 65 times blood plasma volume. Between 178 and 179 litres are returned to the bloodstream, so 1 to 2 litres are excreted as urine. So from our previous session, you'll remember that the nephron consists of a renal corpuscle where the filtration of plasma occurs, and a renal tubule where the filtered fluid collects. The renal corpuscle consists of the glomerulus, and that's the capillary network, alongside the Bowman's capsule. The Bowman's capsule is a double-layered structure that surrounds the glomerulus. The capillary endothelium of the glomerulus and the visceral layer of the Bowman's capsule together make up an endothelial capsular membrane, and this acts as a filter with 3 layers. The first layer is a single layer of capillary endothelial cells, and these have large pores, approximately 50 times more permeable than pores elsewhere. This allows all components of blood plasma to pass through except for the red blood cells. The second layer is a basement membrane of the glomerulus, and this lies between the capillary endothelium and the visceral layer of the Bowman's capsule. It's composed of a mesh of glycoprotein fibrils, and it prevents the filtration of larger proteins. The specialized cells of the visceral layer of the Bowman's capsule are called podocytes, and these form the third filtration layer. Between these podocytes are so-called filtration slits, and these prevent filtration of medium-sized proteins. The difference in pressure between the afferent arteriole and the efferent arteriole produces the filtration pressure, and this forces fluid and solutes into the capsular space within the Bowman's capsule. The volume of blood that's filtered through glomerular capillaries is larger than in other capillaries, and this is due to several factors. Firstly, glomerular capillaries are very long, and this increases the surface area available for filtration. Secondly, the filter is porous and thin, approximately 0.1 micrometers thin, and as we've seen, the pores allow this structure to be 50 times leakier than other capillaries. And lastly, the capillary blood pressure is high. This is because the efferent arteriole, which is leaving the glomerulus, is narrower than the afferent arteriole. Net filtration pressure refers to the total pressure which promotes filtration, and it's dependent upon 3 forces. Firstly, glomerular blood hydrostatic pressure. This is the hydrostatic pressure in the glomerular capillaries, and it's produced by the difference between the afferent and efferent arterioles. This is usually about 55 millimeters of mercury. The second factor is capsular hydrostatic pressure. This is the back pressure in the capsular space due to resistance to the expansion of the walls of the capsule. It's usually about 15 millimeters of mercury. Blood colloid osmotic pressure is the back pressure created by the presence of proteins, for example albumin, globulins and fibrinogen, that cannot pass through the endothelial capsule membrane. As the blood enters the glomerulus, this pressure is about 25 millimeters of mercury, but as it passes through the glomerulus, fluid, solutes, and small to medium-sized proteins pass into the capsular space. The effect of this is to increase that value from 25 to approximately 35. The average blood colloid osmotic pressure within the glomerulus is about 30. So net filtration pressure, i.e. the total pressure promoting filtration, is equal to glomerular blood hydrostatic pressure minus capsular hydrostatic pressure minus blood colloid osmotic pressure. The following link is to a YouTube video which is useful in explaining these different pressures. So just to bring us back, the three major renal processes are glomerular filtration, occurring up in the glomerular capsule, the Bowman's capsule. The second is the tubular reabsorption, which is occurring along the length of the nephron between the tubule and the peritubular capillaries. And the third is tubular secretion, where waste products are secreted into the tubule and excreted from the body as urine. We will now look at the regulation of glomerular filtration rate. Glomerular filtration rate is the amount of filtrate formed in both kidneys per minute, and this is about 125 mils per minute. This process is regulated by adjusting the blood flow into and out of the glomerulus and by altering the glomerular capillary surface that's available for filtration. And there are three mechanisms by which this can be achieved. Firstly, renal autoregulation. The second is hormonal regulation. And thirdly, neural regulation. So this diagram is available from your textbook and shows the process of renal autoregulation. If some stimulus disrupts homeostasis by causing an increased glomerular filtration rate, this will be detected by the macula-densa cells of the juxtaglomerular apparatus. These cells pick up on the increased delivery of sodium, chloride, and water. This information is fed into the control centre in the juxtaglomerular apparatus, and as a result, there is increased secretion of vasoconstrictor. This results in the afferent arteriole constricting, thus decreasing blood flow through the glomerulus. This causes a decrease in glomerular filtration rate, and there will be a return to homeostasis when the response brings glomerular filtration rate back to normal. This mechanism to regulate glomerular filtration rate is called intrinsic because it operates completely within the kidney. The second process is hormonal regulation, and two main hormones are involved in this. The first is angiotensin II. This diagram is available in your textbooks. When blood pressure falls, changes in the reabsorption of sodium ions, chloride ions, and water by the lipid Henle cause the release of the hormone renin by the juxtaglomerular cells. Renin acts upon large plasma protein in the blood called angiotensinogen and removes a small fragment called angiotensin I. This then travels to the lungs where a second enzyme called angiotensin converting enzyme, or ACE, within the pulmonary capillaries converts angiotensin I into the active hormone angiotensin II. All the actions of angiotensin II help to increase the systemic blood pressure and blood volume. It is a potent vasoconstrictor. It constricts afferent arterioles and reduces the renal blood flow, hence decreasing glomerular filtration rate. It will also constrict arterioles elsewhere in the body, thus increasing systemic blood pressure. Angiotensin II stimulates the adrenal cortex to release aldosterone, which increases the reabsorption of sodium, chloride, and water by the kidney, thus increasing blood volume and blood pressure. Angiotensin II also stimulates the thirst centres within the hypothalamus to increase water intake and restore blood volume. It also stimulates the secretion of antidiuretic hormone, ADH, from the posterior pituitary gland. ADH increases reabsorption of water by the kidney. The second hormone that's involved is atrial natriuretic peptide, and this is secreted by cells in the atria of the heart, stimulated by increased stretch in the wall of the heart caused by increased blood volume. ANP increases glomerular filtration rate by dilating afferent arterioles and thus increasing the surface area of the glomerular capillaries. The third process is neural regulation of glomerular filtration rate. Vasoconstrictor fibers from the sympathetic division of the autonomic nervous system are involved in this process. With increased stimulation, both efferent and afferent arterioles will constrict, thus decreasing glomerular filtration rate. With a greater increase in stimulation, increased constriction of afferent arterioles occurs, for example during exercise. Sympathetic stimulation also stimulates the release of renin from the juxtaglomerular cells, and we saw in the previous diagram the renin-angiotensin pathway. So having looked at glomerular filtration, we're going to move on in the next session to look at the second process of tubular reabsorption and the third process of tubular secretion.